There is something quietly extraordinary about the fact that you are here reading this in a body that has lasted as long as it has. Most animals that share your size, your weight, your rough metabolic pace through the world are already gone by the time a human being reaches middle age. A dog that has lived 10 years has lived a full life. A chimpanzee that reaches 40 is considered ancient. A mouse, if it survives its second year, has already outlived most of its kind. And yet a human being at 40 is, by almost every biological measure, still young. A human being at 60 still walks, thinks, learns, laughs, grieves and loves, with the full complexity of a creature that has not yet exhausted its design. A human being at 80, though slowed and worn, carries within every cell of their body a story of adaptation so improbable, so slow and so precise that to understand it is to understand something essential, not only about the species, but about the nature of life itself. This is not a story about medicine. It is not a story about hospitals or vitamins or the modern obsession with longevity that fills bookstore shelves and wellness retreats. This is a much older story. It is the story of how, across millions of years of slow and patient selection, the human body came to age at a pace unlike almost anything else that has ever walked to the surface of this planet. It is a story written not in words but in bone density, in the length of childhood, in the particular way human cells repair themselves, in the extraordinary generosity of grandmothers, in the strange slowness of human development that baffled early researchers and continues to raise quiet, important questions today. And if we approach this story gently, the way one approaches a fire burning low in a dark forest, we begin to see that the way humans age is not simply a biological accident. It is the outcome of a series of ancient decisions made not by individuals, not by tribes, not even by civilizations, but by the blind and patient logic of natural selection working across hundreds of thousands of generations. Every year a human lives beyond what other primates can manage is a year that was, in some ancient environment, worth living. Every decade of relative vitality that human biology affords was, at some point in the deep past, a decade during which something happened that mattered enough to be preserved. To understand this fully, we have to begin not with humans at all. We have to begin with the basic logic of aging itself, which is one of the strangest and most philosophically unsettling facts in all of biology. Because aging, when you think about it carefully, should not exist. It should have been selected against millions of years ago. If an organism could simply live forever, replacing damaged cells, repairing every broken strand of DNA, never accumulating the slow errors that lead to decline, it would reproduce indefinitely, passing its genes forward without limit. And yet almost nothing lives forever. Almost everything ages. Almost everything declines. Almost everything dies. The question of why is one of the deepest in all of evolutionary biology, and the answer, when it comes, is surprising in its simplicity. Aging exists because natural selection does not care about organisms after they have finished reproducing. This is the core of the evolutionary theory of aging, developed in the middle of the 20th century by researchers including Peter Medawar and George Williams, and it is an idea that takes a moment to absorb. From the perspective of natural selection, what matters is not whether an organism lives a long life, but whether it passes its genes successfully to the next generation. Once reproduction is complete, once offspring are raised and dispersed, the organism itself has, in a sense, served its purpose. Any mutations that cause damage or decline after that point face almost no selective pressure. they may accumulate quietly, generation after generation, without being removed. And so aging in this view is not a design, but an absence of design. It is what happens when selection stops paying attention. But this framing, elegant as it is, immediately raises a further question. If aging is what happens when selection stops caring, then why do different species age so differently? Why does a mouse decline and die within 2 or 3 years, while a Greenland shark can live for nearly 500? Why does a small songbird age faster than a large parrot? Why does a chimpanzee, our closest living relative, begin to show serious signs of physical decline in its 30s, while a human being of the same age is frequently in the prime of life? The answer lies not simply in size, not simply in metabolic rate, not simply in any single factor, but in the particular ecological circumstances that each species evolved within, and the particular reproductive strategies that those circumstances demanded. This is where the human story becomes genuinely remarkable. Because humans did not simply evolve to live longer by accident. They evolved to live longer because, in the particular ecological niche that early hominins came to occupy, living longer was extraordinarily useful, and the path to that longer life was paved not with comfort, but with challenge, not with safety, but with extraordinary difficulty, and not with individual advantage alone, but with something that took researchers a long time to recognise as a biological force of enormous power, the advantage of older individuals to the group as a whole. To understand how this worked, we need to travel back in time, not to the prehistoric past in any vague sense, but to a very specific period. Roughly 2 million years ago, when a lineage of hominins in Africa began doing something that no other primate had done before. They began to expand the range of foods they could eat by processing those foods before eating them. At first, this processing was mechanical, the smashing of bones to access marrow, the pounding of tough plant material against flat stones. Later, and this is one of the most consequential moments in the history of life on Earth, it involved fire. The controlled use of fire to cook food, which researchers now believe may have begun somewhere between 1 and 2 million years ago, changed almost everything about human pathology and human social life. And one of the things it changed quietly and over many generations was the pace at which humans aged. Cooking increases the caloric yield of almost every food. It breaks down tough fibers, denatures proteins, gelatinizes starches, and kills many of the pathogens that would otherwise cause illness. A cooked sweet potato provides significantly more usable energy than a raw one. Cooked meat is far easier to digest than raw meat. When early hominins began cooking, they were not simply changing their diet. They were fundamentally increasing the amount of energy available to their bodies, and that surplus of energy had to go somewhere. Some of it went into larger brains. Some of it went into the support of the longer childhoods, and some of it went, in ways that are still being worked out by researchers, into the maintenance and repair of the body itself. Processes that are enormously expensive in biological terms, and that only become viable when there is a consistent surplus of energy to pay for them. This connection between energy surplus and the slowing of aging is not obvious, but it is deeply embedded in the biology of all living things. Every organism faces what biologists sometimes call the life history trade-off. The fundamental tension between the resources invested in current reproduction and the resources invested in survival and maintenance for future reproduction. An organism that invests everything in producing offspring quickly will do well, when environments are dangerous and unpredictable, because it will not be alive long enough to benefit from saving energy for later. But an organism that invests heavily in its own maintenance, repairing cells, rebuilding tissues, protecting against oxidative damage, will survive longer and reproduce across a longer window of time. The question of which strategy is better depends entirely on the environment. In the environments that shaped the earliest members of our genus, the environment of the African savanna roughly 2 million years ago, both strategies were simultaneously under pressure. The world was dangerous. Predators were common. Resources were uncertain. The infant mortality rate was, by any modern standard, catastrophic. Most individuals who were born did not survive their first few years, and yet despite all of this, something was pushing the human lineage toward a greater longevity rather than simply faster reproduction. Something was making it worthwhile for a human body to invest heavily in its own maintenance, to slow its own pace of decline, to persist long past the age at which most primates were already worn out. That something, researchers now believe, was the extraordinary complexity of the environment that early humans were navigating. A chimpanzee, which is a sophisticated animal by any measure, learns what it needs to know to survive largely through individual exploration and imitation over the course of a few years of childhood. It is sexually mature by age 10 or 11, and it is largely independent from its social group long before that. But a human child, in the conditions under which the species evolved, required an extraordinary length of time to acquire the knowledge and skills necessary to function as an adult. Hunter-gatherer children in surviving traditional societies typically do not become effective hunters or foragers until their late teens or early 20s. Before that, they require enormous amounts of care, feeding, teaching, and social support, and the investment required to raise a single human child to competence is so large that it has no parallel in any other primate. This creates an immediate and obvious problem. If raising a single human child requires 15 or 20 years of sustained investment, and if the child cannot feed itself for most of that time, then the reproductive success of any individual human depends not only on the survival of the child, but on the maintenance of the entire support network that sustains the child. And this is where the aging of the body becomes deeply entangled with the aging of the group. Because in a small band of hunter-gatherers, the loss of any experienced adult member is not simply a personal tragedy. It is a disruption to the entire web of knowledge, skill, and social connection that keeps everyone alive. Every experienced forager who dies takes with them a map of the landscape, an understanding of our seasonal patterns, a knowledge of which plants are edible in a drought year, which animal trails lead to water, which roots can serve as medicine, which berries will kill you, and which will restore your strength. That knowledge, accumulated over decades of a life experience, is not written down. It exists only in the minds and hands of the people who have lived long enough to gather it. This is the ecological context in which the extraordinary human lifespan began to evolve, and it's a context that makes the evolution of slower aging not merely plausible, but almost inevitable. Because in any group where experienced older individuals contribute meaningfully to the survival of younger ones, selection will favour the survival of those older individuals. Mutations that promote longevity, that slow the pace of cellular decline, that allow the body to maintain itself longer than the bodies of other primates, will persist in the population because the individuals who carry them will live longer, and because the groups that contain those individuals will fare better. The logic is circular in the way that the most powerful evolutionary forces tend to be. Longer life makes the group stronger, and stronger groups support longer lives. But there is a further dimension to this story that took researchers a long time to appreciate, partly because it required taking seriously something that had been overlooked for most of the history of evolutionary biology. For most of the 20th century, when researchers thought about the evolution of longevity, they thought primarily about the survival and reproductive success of individual organisms. The question was always framed in terms of what was good for the individual, what traits a given individual needed in order to pass its own genes to the next generation. This framing is powerful and largely correct, but it missed something important about the human case. It missed the extraordinary degree to which human reproductive success is dependent not on individual effort alone, but on the contributions of others who are not currently reproducing. And in particular, it missed the contributions of older women. In almost every other primate species, females remain reproductively active until the end of their lives. They do not experience menopause or anything like it. When they can no longer successfully reproduce, they're also nearing the end of their biological capacity to do much else. But human females routinely live for decades after their last pregnancy, decades during which they are by any measure still biologically active, still physically capable, still cognitively sharp. For a long time, this was regarded as something of a biological puzzle, a developmental leftover with no particular adaptive significance. It seemed wasteful, from an evolutionary perspective, for an organism to invest in maintaining a body that was no longer reproductively active. Then, in the 1990s, a researcher named Kristen Hawkes and her colleagues began a series of detailed observations of the Hadza people of Tanzania, one of the last remaining hunter-gatherer populations in the world. And what they found changed the way scientists think about human aging. They found that older Hadza women who are long past menopause, were among the most productive foragers in the entire group. They were spending more hours gathering food than anyone else, and the food they gathered was going not to themselves, but disproportionately to their grandchildren, and to the children of other group members. These grandmothers were, in effect, subsidizing the reproduction of the next generation. They were enabling younger women to wean their children earlier than would otherwise be possible, because the nutritional shortfall of early weaning was being made up by the contributions of older women who no longer needed to invest energy in their own reproduction. This observation gave rise to what is now known as the grandmother hypothesis, one of the most significant and debated ideas in the study of human evolution. The hypothesis proposes that the evolution of menopause in human females, rather than being a biological accident or a side effect of some other change, was an adaptive strategy that allowed older women to shift their reproductive investment from producing new offspring of their own to supporting the survival and development of their existing offspring and grandchildren. And because this strategy was so effective at increasing the reproductive success of close relatives, it created strong selection pressure for the survival of older women, which in turn created selection pressure for the maintenance of the body well past the age of reproduction. In other words, grandmothers helped create the conditions that made human longevity possible. The implications of this idea extend far beyond the specific question of menopause. They suggest that the evolution of the human lifespan was not driven simply by the survival of the individual who carries the genes, but by the survival and flourishing of the kinship network within which that individual lives. The human body, in this view, is designed not for an individual life, but for a social life. and its lifespan reflects not simply how long an individual needs to survive to reproduce, but how long an individual can remain useful to the web of relationships that sustains everyone around them. This is a deeply different way of thinking about aging than the one we typically encounter in modern discussions of longevity, and it is in many ways a more human one. But the grandmother hypothesis is only one strand of a much larger story. To understand why humans age more slowly than other animals, we also need to look at the body itself, at what is happening at the level of cells and tissues and molecules when one organism ages more gracefully than another. Because the slowing of aging is not a single change, it is the outcome of dozens of interrelated biological adjustments, each of which had to evolve gradually, each of which imposed its own costs and required its own ecological justification. And when we look at these adjustments closely, we find in them a picture of extraordinary biological ingenuity, not the ingenuity of conscious design, but the ingenuity of selection working on variation across millions of years with patience that no human mind can fully comprehend. One of the most fundamental mechanisms of aging in any organism is the gradual accumulation of damage in the genetic material of cells. Every time a cell divides, it must copy its entire genome, the complete set of instructions encoded in its DNA. And every time this copying happens, there is a chance of error. Most of these errors are caught and corrected by the cell's own repair machinery, which is extraordinarily sophisticated. But not all errors are caught. Some slip through, and over the course of many years and many cell divisions, these accumulated errors begin to interfere with the normal functioning of cells and tissues. This process is one of the primary drivers of the physiological decline we call aging. At the ends of every chromosome in every cell sits a structure called a telomere, a repetitive sequence of DNA that serves as a kind of protective cap, preventing the chromosome from degrading or fusing with neighbouring chromosomes. Every time a cell divides, the telomere at the end of each chromosome gets slightly shorter, because the molecular machinery that copies DNA cannot quite reach the very end of the strand. Eventually, after enough divisions, the telomere becomes critically short, and the cell either stops dividing entirely, entering a state called senescence, or it dies. This process of telomere shortening is one of the primary biological clocks that governs the lifespan of cells, and by extension, the lifespan of the organisms those cells compose. What is remarkable about humans in comparison with shorter-lived primates is not that humans have especially long telomeres to begin with, though there is some variation in this, but that the human genome encodes a more sophisticated set of tools for managing the damage that accumulates in cells over time. Human cells express, at higher levels than chimpanzee cells, a variety of enzymes and regulatory proteins involved in DNA repair, in the management of oxidative stress, in the clearance of damaged proteins, and in the regulation of inflammation. These are not dramatic differences. They are subtle shifts in the activity of molecular pathways that have ancient evolutionary origins shared across all of mammals. But small differences in the efficiency of these pathways, multiplied across trillions of cells and billions of cell divisions over the course of a lifetime, add up to very large differences in the pace of biological decline. One of the most important of these pathways involves the response to oxidative stress. Every cell in your body, as part of its normal metabolic activity, generates highly reactive molecules called reactive oxygen species, a byproduct of the process by which cells extract energy from food. These molecules are chemically aggressive, and they damage proteins, lipids, and DNA wherever they encounter them. The accumulation of this oxidative damage is one of the primary drivers of aging in all aerobic organisms, and cells have an elaborate system of antioxidant enzymes, molecules like superoxide dismutase and ketolase, that neutralize reactive oxygen species before they can cause damage. The efficiency of this system varies between species, and it varies in ways that correlate strongly with lifespan. Long-lived animals tend to produce reactive oxygen species at a lower rate, and to neutralize them more efficiently than short-lived animals of comparable size. This is not simply because they have bigger bodies or slower metabolisms, though both of those factors play a role. It is because their genomes encode versions of these protective enzymes that are either more active, more numerous, or more precisely regulated than those of their shorter-lived relatives. And in humans, this pattern reaches what is, among primates, an unusual extreme. The human genome encodes a particularly robust set of antioxidant and D-Candor-1A repair pathways, and these pathways are expressed in patterns that favour longevity over the kind of fast, intense reproductive output that characterises many shorter-lived mammals. This brings us to one of the most fundamental tensions in the biology of aging, a tension that George Williams articulated in the 1950s in an idea he called antagonistic pleiotropy. Williams noticed that many of the genes that promote vigorous growth and reproduction in young animals have negative effects on survival later in life. A gene that promotes rapid cellular proliferation in a young organism may also increase the risk of cancer in an old one. A gene that promotes an aggressive immune response to infection in youth may contribute to chronic inflammation in old age. A gene that promotes the mobilization of calcium from bone during periods of nutritional stress may increase the risk of osteoporosis in later life. These are not design flaws. They are the inevitable outcome of selection that acts much more powerfully on young organisms than on old ones, and that therefore tolerates negative effects later in life, if those negative effects are the price of vigorous performance early on. What makes human aging unusual in this framework is that the costs of antagonistic pleiotropy appear to be somewhat delayed in humans relative to other primates. The trade-offs that in a chimpanzee begin to manifest in the third decade of life do not typically become apparent in humans until the fifth or sixth decade. This is not because humans have somehow escaped the fundamental logic of antagonistic pleiotropy, but because the extraordinary value of experienced adults in human social groups created selection pressure for pushing those costs further into the future. Every decade of additional vitality that natural selection could purchase, at whatever biological cost, was a decade during which a knowledgeable elder could continue contributing to the group's survival. And so, the costs were deferred year by year, generation by generation, across hundreds of thousands of years of selection, until the human body had become something genuinely unusual in the history of primate evolution, an organism that remains capable and contributing well past the age at which its closest relatives are already in serious decline. But we should be careful not to describe this process in a way that makes it sound clean or simple or directed. Evolution does not have a goal. Natural selection does not know where it is going. What happened over the course of human evolutionary history was not that some ancient wisdom chose to invest in longevity. It was that, in the particular social and ecological circumstances of early human life, individuals who happened to carry genetic variants that promoted slightly longer survival happened to contribute more to the survival of their relatives, and those relatives survived to carry the same variants forward. This happened not once or twice. but across an almost incomprehensible number of generations, with countless small adjustments and countless failed experiments along the way, until the human genome had accumulated a particular set of characteristics that, taken together, produced the pattern of aging we observe today. One of the most striking of these characteristics, one that rarely makes its way into popular discussions of human longevity, is the extraordinary length of human childhood. Among all primates, and indeed among all mammals, humans have the longest period of developmental dependency. A chimpanzee infant is born helpless, like all primate infants, but it develops far faster than a human child. By the age of 3 or 4, a young chimpanzee has learned many of the basic skills it will need as an adult. By the age of 8 or 9, it is largely capable of feeding itself. By 10 or 11, it has reached sexual maturity and can begin reproducing. The entire developmental trajectory from birth to reproductive adulthood takes roughly a decade. In humans, this trajectory is almost doubled. Human children are helpless for far longer than chimpanzee infants. They develop cognitive and motor skills more slowly, not because they are less capable, but because their brains are doing something far more complex, building a cognitive architecture that will eventually be capable of language, abstract reasoning, social manipulation, long-term planning, and the kind of cultural transmission that allows knowledge to accumulate across generations. This takes time, a great deal of time, and during all that time, the child must be fed, protected, and taught by others, primarily by adults who have themselves survived long enough to accumulate the experience and knowledge that make them effective teachers. The connection between extended childhood and extended lifespan is not accidental. It is deeply causal. A species that requires 15 or 20 years to produce a reproductively competent adult cannot afford to lose the experienced adults who guide that development before the process is complete. And so selection that extended childhood simultaneously created selection pressure for extended adult lifespan. The two evolved together, each pulling the other forward, until the human life history had been stretched in both directions, a longer childhood on one end and a longer post-reproductive lifespan on the other, with a window of reproductive activity in the middle that is actually not dramatically longer than that of other primates. What is unique about humans is not primarily the length of the reproductive period itself, but the extraordinary extensions at both ends. If we look deeper at the biological mechanisms that support this extended human lifespan, we find a collection of adaptations that are, in their individual details, relatively modest, but that in combination produce something remarkable. The human immune system, for example, is slightly more sophisticated in certain respects than that of shorter-lived primates, with a somewhat greater capacity for adaptive immunity, the kind of immune response that can remember past infections and respond to them more rapidly in the future. This matters for longevity because infectious diseases across the lifespan, one of the primary sources of cellular and systemic damage that contributes to aging. An immune system that manages infectious threats more efficiently throughout life will allow the rest of the body's systems to age more slowly, simply because they're not constantly dealing with the aftermath of uncontrolled infection. The human gut microbiome, which has only recently begun to receive the scientific attention it deserves, appears to play a role in healthy aging that researchers are only beginning to understand. The collection of microorganisms that inhabits the human gastrointestinal tract is not simply a passive presence. It actively participates in the metabolism of food, in the regulation of the immune system, in the production of certain vitamins and signaling molecules, and important in ways that are still being elucidated in the regulation of systemic inflammation. Chronic low-grade inflammation is one of the primary molecular drivers of aging in all mammals, and anything that reduces the baseline level of systemic inflammation will, over time, slow the pace of biological decline. The human gut microbiome, shaped by millions of years of coevolution with its host, appears to be tuned in ways that support this kind of anti-inflammatory modulation, at least when it is provided with the kind of diet that shaped it during evolution. This brings us to one of the most important, most frequently misunderstood dimensions of the human aging story. The extraordinary lifespan that human biology makes possible, the potential for 8 or 9 decades of life that is encoded in the human genome, was not designed for the world we live in today. It was designed for a world that no longer exists. It was designed for the world of the hunter-gatherer, a world characterized by constant, moderate physical activity, a diet of wild plants and animals with enormous variety and no refined carbohydrates, a social life embedded in a small intimate group of people who knew each other deeply, a sleep pattern governed by natural light cycles rather than electric illumination, and a relationship with the physical environment that involved daily exposure to sunlight, fresh air, variable temperature, and the microbiological richness of soil and plants and water. In that world, the biological machinery for slow aging worked as it was designed to work. The repair mechanisms were activated appropriately by the right kinds of stress at the right intensities. The inflammatory response was triggered by actual threat and resolved promptly when those threats passed. The gut microbiome was maintained in its ancient diversity by the enormous variety of plant foods that hunter-gatherers consumed. The telomere-shortening clock of cellular aging was calibrated to an environment in which the body was stressed in moderate ways that actually promoted cellular resilience, not overwhelmed by sedentary excess that allowed damage to accumulate without the stimulation of repair. The modern world has changed almost every one of these parameters in ways that the human genome did not anticipate. We eat a fraction of the dietary variety that shaped our digestive systems over millions of years. We move far less than our bodies were designed to move. We sleep in the presence of artificial light that disrupts the hormonal rhythms that govern cellular repair and immune function. We experience forms of stress that are psychological rather than physical, chronic rather than acute, and for which the fight or flight response that evolution provided us is not well suited. And we live in environments of extraordinary hygienic cleanliness that have eliminated many of the microbial exposures that shaped our immune systems and may have been important for their calibration. The result of all these mismatches is that the human potential for slow aging is rarely fully realized in modern conditions. The body that evolution built for longevity in the African savanna is now trying to navigate an environment of supermarkets, office chairs, electric lights, and social media, and it is doing so with a set of biological tools that were forged over hundreds of thousands of years for a very different world. This is not cause for despair, it is cause for understanding. Because understanding the evolutionary origins of human aging is one of the most powerful tools we have for understanding what the body actually needs in order to age as slowly as it was designed to age. To appreciate this more fully, it helps to look at how populations that live in conditions close to those of our evolutionary past actually age. Research on hunter-gatherer and traditional subsistence populations around the world has consistently found that while these populations face very high mortality from infectious disease and injury, particularly in childhood, The individuals who survive into adulthood often maintain impressive physical and cognitive function into old age. Traditional foraging populations show very low rates of the chronic diseases that dominate modern medicine, including cardiovascular disease, type 2 diabetes, many cancers, and many forms of cognitive decline. Their bodies, engaged in daily moderate activity, eating an enormously varied diet, sleeping in natural light cycles, and living embedded in dense social networks, age in ways that more closely approximate what the human genome was designed to support. This does not mean that life in traditional societies was idyllic, or that hunter-gatherers were somehow more authentic or superior human beings. Their lives were hard in ways that are difficult for modern people to fully appreciate. They experienced pain, loss, and deprivation at rates that would be intolerable by contemporary standards. But what their experience does reveal is something important about the relationship between the human body and the world it evolves it in. A relationship that was, for most of human history, a relationship of genuine fit, in which the demands of the environment and the capabilities of the body were matched to each other in ways that allowed the full potential of human biology to express itself. From an evolutionary perspective, the key insight is this: the human lifespan was not given to us by chance. It was earned across millions of years of selection by the accumulating evidence that long-lived humans were more useful to the groups they lived in than short-lived ones. The knowledge they carried, the support they provided, the grandchildren they fed and protected and taught, the stability they contributed to the social fabric of small bands navigating an unpredictable and often dangerous world. All of these things translated generation by generation into slightly stronger selection for the biological mechanisms that keep the human body functioning longer than the bodies of its evolutionary relatives. And when we ask what those mechanisms are, specifically, the answer turns out to be a fascinating collection of biological innovations that humanity shares with no other species in quite the same combination. Some of these innovations are genetic, encoded in the particular variants of repair and maintenance genes that are common in human populations. Some are developmental, expressed in the extraordinary slowness and complexity of human growth and maturation. Some are social, depending on the existence of stable, knowledge-rich social groups in which experienced individuals can contribute meaningfully over long periods of time. And some are cultural, preserved in the accumulated practices and traditions by which human groups have always sought to maintain the health and vitality of their members across the full arc of life. It is worth pausing here to consider what it means that human longevity is, in this sense, a social achievement, rather than simply a biological one. We are accustomed to thinking of aging as something that happens inside individual bodies, a private decline that medicine may be able to slow, but that is ultimately the business of cells and tissues and molecules. But the evolutionary story of human aging suggests something different. It suggests that the human body was designed for a particular kind of social environment, one in which older individuals are valued and supported, in which the knowledge and experience of elders is actively sought out and incorporated into the life of the group, and in which the biological investment required to keep an older body functioning is matched by a social investment in ensuring the older person has access to adequate nutrition, support, and purpose. In the environments in which human longevity evolved, these social conditions were largely self-maintaining. In a small band of hunter-gatherers, every member's contribution was visible and necessary. An experienced elder who could identify edible plants in a drought year, who could track animal movements across a landscape they had known for decades, who could mediate conflicts before they tore the group apart, who could tell the stories that kept the group's history and values alive across generations, was obviously and immediately valuable. There was no need for an abstract argument about the value of older people. Their value was tangible and daily. In the modern world, these conditions are far less guaranteed. The social fabric within which human longevity evolved has been largely dissolved by the forces of industrialization, urbanization, and individualism. Many older people in modern societies lack the dense social networks and the sense of meaningful contribution that appear to be important not only for psychological well-being, but for physical health and longevity itself. Research consistently finds that social isolation is one of the strongest predictors of early mortality in older adults, with effects on lifespan comparable in magnitude to those of smoking. This finding makes a great deal of sense in light of the evolutionary story we have been exploring. The human body was designed to age slowly within a social context. When that context is removed, the biological machinery of longevity loses one of its most important inputs. But this is not the only way in which the modern world has created a mismatch with the conditions that shaped human aging. There is another dimension of this story, one that is less often discussed, and it involves a relationship between the pace of life and the pace of aging. The pace at which a body ages is not fixed at birth. It is modulated throughout life by the conditions the body experiences in ways that reflect the extraordinary plasticity of biological systems. The human body is not a clock that simply runs down at a predetermined rate. It is a dynamic system that is constantly adjusting its own maintenance programs in response to the signals it receives from the environment. One of the most important of those signals is the level and nature of physical activity. The human body evolved in an environment of consistent, moderate, varied physical activity. Hunter-gatherer populations that have been studied move an average of 9 to 15 kilometers per day, primarily walking, with periods of more intense activity interspersed. This level of activity activates a broad suite of biological maintenance programs, stimulates the production of proteins that promote cellular repair, reduces systemic inflammation, improves the efficiency of mitochondria, the energy-producing organelles of cells, and modulates the hormonal environment of the body in ways that slow the accumulation of the damage that leads to aging. When the body does not receive this signal, when it is sedentary day after day in the way that modern life encourages, these maintenance programs are not activated at the same intensity. The biological machinery of repair and maintenance shifts toward a lower gear, and the result over time is a pace of aging that is faster than what the human genome was designed to support. This is not a moral failing, it is a physiological one, a mismatch between the body's expectations and the world it actually inhabits. But it is a mismatch with real consequences, consequences that are now being carefully quantified by researchers studying the biology of aging in different populations and in controlled exercise experiments. The relationship between diet and aging pace is similarly complex and similarly mismatched with modern conditions. The human digestive system evolved over millions of years in an environment in which the diversity of plant foods consumed in a year might number in the hundreds of species. Different fruits, leaves, roots, tubers, seeds, nuts, and fungi, each containing unique combinations of fiber types, polyphenols, vitamins, and other bioactive compounds, presented the gut and its microbial inhabitants with a constantly varying menu that maintained the diversity and function of the microbiome. and provided the raw materials for the body's many repair and maintenance processes. Modern agricultural diets, and especially the ultra-processed versions of those diets that have become dominant in industrialized societies, provide a tiny fraction of this diversity. The average American eats food derived from perhaps a dozen plant species. The variety of fiber types available to the gut microbiome has collapsed dramatically. Many of the polyphenolic compounds that served as biological signals to the body's maintenance systems, compounds that in an evolutionary context were available in some quantity in almost every meal, are now largely absent from the diet. The result is a gut microbiome that is less diverse and less functional than it was for most of human evolutionary history, with consequences for inflammation, immune function, and ultimately for the pace of aging. None of this is to suggest that modern humans are doomed to age faster than their ancestors. The biological machinery of slow aging is still present in the human genome, ready to respond to the right inputs. Research on the effect of dietary diversity, physical activity, social connection, sleep quality, and exposure to natural environments on the pace of biological aging has consistently found that these factors can shift the body's aging trajectory in meaningful ways, not dramatically, not magically, but in ways that are real and measurable and consistent with what the evolutionary story predicts. The human body is not a machine that wears out at a fixed rate. It is a living system that responds to its environment, and it responds within limits in the direction that the environment invites it to go. This is one of the most important messages embedded in the evolutionary story of human aging. We did not evolve to age slowly because our ancestors were special, or because they were trying to live long lives. They were not thinking about longevity. They were thinking, to the extent that early hominins were thinking about anything in the long-term sense, about immediate survival. Food, water, safety, warmth, the care of the young, the maintenance of social bonds, the navigation of a complex and dangerous world with limited resources and just enormous intelligence. Longevity was a byproduct of all of this, a consequence of the particular way that human social and cognitive complexity created an environment in which older individuals were genuinely, demonstrably, quantifiably valuable. The lesson is not that we should try to return to the lives of hunter-gatherers, or that modern medicine and technology are somehow misguided. The lesson is subtler and more interesting than that. It is that the human body contains within it a biological potential for long, relatively healthy aging that is not automatically expressed in every environment, but that can be more or less fully realized depending on how closely the conditions of life approximate the conditions that shaped that potential over evolutionary time. And understanding those conditions, understanding what the body actually expects and needs from its environment, is perhaps the most powerful tool we have for living well across the full arc of a human life. There is something deeply moving about this story when you sit with it quietly and let it unfold at its own pace. Each of us, the temporary custodians of a biological inheritance that has forged across millions of years of struggle and adaptation. Every mechanism in our bodies that allows us to repair a damaged cell, to neutralize a toxic molecule, to maintain the integrity of our genetic material through another decade of living, was earned by ancestors who lived and died in circumstances we can barely imagine. They did not know they were contributing to our longevity. They did not know they were part of a story that would eventually produce creatures capable of understanding the story. They simply lived as fully and as long as their bodies and their worlds allowed them to, and in doing so they left us this extraordinary inheritance. The pace of our aging is not a fixed destiny. It is a conversation between our biology and our world, a conversation that has been going on for millions of years and that we are still, in a very real sense, participating in every day. When we move, when we eat with variety and care, when we sleep in accordance with the rhythms our bodies were shaped to expect, when we maintain the social bonds that evolution built us to need, when we find meaning and purpose and contribution in our communities, we're not simply following health advice. We are, in some small way, continuing a conversation with our own deep past, honouring the investment that millions of years of selection made in these bodies, and giving the extraordinary biological machinery of human longevity the conditions it was built to work in. And when we look at an older person who has remained curious, engaged, generous, and physically capable well into the later decades of life, we are seeing something that is not simply the product of good luck or good genes, though both of those play a role. We are seeing the expression of one of the most remarkable achievements in the history of life on Earth. The slow, patient, improbable evolution of a body designed to keep contributing, keep learning, keep connecting, keep caring, across a span of time that has no parallel in the primate world. This is the story of how we came to age more slowly than other animals. Not because we were designed for comfort or safety, not because we escaped the pressures of natural selection, but because we evolved in conditions where staying alive, staying sharp, staying connected, and staying useful for as long as possible was the most powerful thing a body could do for the people it loved and the world it lived in. And perhaps, if we are quiet enough to hear it, that is still true. The night is long, and the world is patient, and the body you carry into sleep's night is older than you can imagine, and wiser, and more deeply designed for the act of living than any of us fully appreciate. Rest now. Let the old machinery do its work. To understand the full depth of why humans age more slowly than other animals, it helps to sit with the numbers for a moment. Not the cold statistical numbers of a laboratory report, but the quiet, astonishing numbers of geological time. The lineage that eventually gave rise to modern humans diverged from the lineage that gave rise to modern chimpanzees somewhere between 6 and 7 million years ago. This is a span of time so vast that it resists intuitive comprehension. 6 million years. If you try to count to 6 million, one number per second, it would take you nearly 70 days without stopping. If you tried to represent it in human generations, assuming a generation of roughly 25 years, it would be 240,000 generations. 240,000 generations of living, dying, reproducing, and being selected upon. 240,000 generations of tiny variations between individuals, some of which made their carriers slightly more likely to survive and reproduce, some of which made them slightly less likely, and most of which made no difference at all. Across these 240,000 generations, the genome of the human lineage was shaped, molecule by molecule, variant by variant, into something that looks increasingly different from the genome of our closest evolutionary relatives. The genetic distance between humans and chimpanzees is sometimes described as small, and in absolute terms it is. Humans and chimpanzees share approximately 98.7% of their protein-coding DNA, but that 1.3% distributed across a genome of roughly 3 billion base pairs amounts to an enormous number of individual differences, and many of those differences are not in genes themselves but in the regulatory sequences that control when, where, and at what level genes are expressed. Changes in gene regulation can have profound effects on the biology of an organism without changing the genes themselves, and it is increasingly clear that many of the most important differences between humans and chimpanzees, including differences in brain development, in immune function, in metabolism, and in the pace of aging, are driven at least in part by regulatory evolution rather than by changes in the protein coding sequences of genes themselves. This regulatory evolution is one of the reasons that the story of human aging is so difficult to tell at the level of individual molecules, and still harder to communicate in popular terms. There is no single gene, no single mutation, no single molecular innovation that makes humans age more slowly than chimpanzees. There are instead hundreds or thousands of small regulatory changes, each of which slightly adjusts the expression of some gene involved some aspect of cellular maintenance, repair, or defense against oxidative damage, and whose cumulative effect over the course of a lifetime adds up to the difference between a chimpanzee that is biologically old at 35 and a human who's biologically middle-aged at 50. This kind of distributed polygenic evolution is the hardest kind to study and the hardest kind to describe, but it is also the most typical kind, and understanding that it is typical is itself an important insight into how evolution actually works. One of the most illuminating comparative studies of human and chimpanzee aging was published in the early years of the 21st century, when researchers began using high-throughput genomic technologies to compare gene expression patterns in the tissues of living humans and chimpanzees at different stages of life. What they found was that while many genes are expressed at similar levels in the two species, at equivalent life stages, there are systematic differences in the expression of genes involved in inflammation, in mitochondrial function, and in the response to cellular stress. In humans, genes that promote anti-inflammatory responses tend to remain more active to later life than their counterparts in chimpanzees. Genes involved in mitochondrial efficiency, which matters for aging because mitochondria are both the primary source of reactive oxygen species and the primary location of oxidative damage, show regulatory patterns in humans that appear to support more sustained mitochondrial function across a longer lifespan. And genes involved in the cellular stress response, the molecular machinery that cells activate when they are damaged or under threat, show patterns of expression in humans that maintain higher baseline activity levels well into middle age compared with chimpanzees of equivalent biological maturity. These are not dramatic differences in isolation, but they are consistent differences, differences that point in the same direction across multiple biological systems, and their consistency suggests that they are not random. They suggest instead that there was sustained selection pressure across many generations of early human evolution for exactly this kind of distributed enhancement of maintenance and repair across multiple biological systems simultaneously. And when we ask what that selection pressure was, we come back to the same answer we found before. It was the extraordinary value in the particular social and ecological context of early human life of experienced individuals who'd survived long enough to accumulate knowledge, skills, and social relationships that benefited everyone around them. In early human environments, information was the most valuable resource of all, more valuable than any individual food item, more valuable than any particular tool, more valuable than any temporary advantage in physical strength or speed, was the accumulated knowledge of someone who had lived through many seasons, many droughts, many migrations, many conflicts, and had retained and integrated and organized that experience into a form that could be communicated to others. In the environment of evolutionary adaptation, before writing, before the internet, before libraries, before any of the external memory systems that modern humans take for granted, all knowledge existed only in the minds of the people who had learned it, and the death of a knowledgeable person was an irreplaceable loss of information. This creates a selection dynamic that is unusual in the animal kingdom, and that has no close parallel even among our nearest relatives. In most animal species, the death of an individual, however capable, does not result in a significant loss of information to the group, because most of what the individual knew was not uniquely valuable, was not communicated to others, and was not the kind of complex, contextual, experience-based knowledge that takes decades to develop. But in human groups, the death of an elder was a genuine informational catastrophe. It might mean that the group no longer had anyone who knew where a particular water source could be found in a drought year, or who understood the social dynamics of a neighbouring group well enough to navigate a potential conflict, or who could identify the particular subtle signs that a particular plant was toxic at a particular season but safe at another, or who knew the stories that preserved the group's history and identity, and explained why certain practices and taboos existed. The loss of any one of these knowledge stores could, in a bad year, mean the difference between survival and starvation, between peace and war, between the maintenance of a crucial social alliance and its collapse, and so selection favoured strongly and consistently any variant that allowed an experienced adult to remain alive and functional for a few more years. Not dramatically longer, not in ways that required miraculous biological innovation, but just a little longer, just enough to pass on one more season's worth of knowledge, just enough to train one more young forager, just enough to help one more grandchild survive its first dangerous years. And here is the deep irony at the heart of this story. The very cognitive complexity that made human knowledge so valuable, and that therefore created such strong selection for longevity, was itself one of the primary drivers of the extended childhood that made longevity so necessary in the first place. The human brain is the most metabolically expensive organ in the known universe relative to its size. It consumes roughly 20% of the body's entire energy budget, even at rest. Building it takes an extraordinary amount of time and resources. The human brain at birth is only about 25% of its adult volume, far less developed relative to its eventual adult size than the brains of any other primate at birth, and it continues growing and reorganizing itself for the first 2 to 3 decades of life, well past the onset of sexual maturity. This prolonged brain development is both what makes human cognition so powerful and what makes human childhood so long. The extended period of brain development requires, among other things, extended periods of sleep, especially the particular phases of sleep associated with memory consolidation and synaptic pruning. Children sleep more than adults. Adolescents, despite the cultural stereotype of laziness, appear to have a genuine biological need for more sleep than adults, related to the particularly intense phase of neural reorganization that occurs during adolescence, and older adults, whose brains are no longer growing, but are still performing the nightly maintenance routines of memory consolidation and cellular repair that sleep makes possible, continue to require substantial sleep well into old age, though the architecture of that sleep changes in ways that are only beginning to be understood. The relationship between sleep and aging is one of the most rapidly developing areas in the biology of longevity. Research over the past 2 decades has established that sleep is not simply a period of rest and inactivity. It is a period of extraordinarily intense biological activity, during which the brain flushes out that of the metabolic, so waste products that accumulate during waking activity, including proteins like beta amyloid that are implicated in neurodegenerative diseases. This flushing process, which occurs through a system of channels in the brain that was only discovered in the early 21st century and is called the glymphatic system, appears to be one of the primary mechanisms by which the brain maintains its own health across a long lifespan. Animals whose sleep is chronically disrupted accumulate these waste products faster and show signs of neurodegeneration earlier than animals whose sleep is protected. This finding connects the story of human longevity to the story of human sleep in a way that is both scientifically interesting and evolutionarily illuminating. The extraordinarily long human lifespan, and particularly the maintenance of cognitive function, across that long lifespan appears to depend critically on the regular, adequate activation of the glymphatic system during sleep, and the conditions under which this system works best: consistent sleep duration, appropriate alignment with natural light cycles, freedom from chronic stress that disrupts sleep architecture, are conditions that were largely present in the evolutionary past and are frequently absent from the modern world. This is yet another dimension of the broader pattern we've been observing throughout this story. The human body's extraordinary potential for slow aging was designed for a particular set of environmental conditions, and when those conditions are absent, the potential is not fully realized. The biology of human sleep is itself a reflection of the social character of human evolution. Humans, like most social primates, did not historically sleep alone. They slept in groups, in close physical proximity to other members of their band, and this social sleeping arrangement served multiple functions simultaneously. It provided warmth through shared body heat, an important consideration in the variable temperatures of the African savanna, especially at night. It provided collective vigilance, since a group of sleeping individuals is harder to approach undetected than a solitary one, and since any one member's arousal in response to a perceived threat would alert the entire group. And it provided the hormonal benefits of social proximity, including the release of oxytocin associated with physical closeness, which modulates the stress response and promotes the kind of deep, restorative sleep phases during which cellular repair and memory consolidation occur most efficiently. When we look at the sleep patterns of remaining hunter-gatherer populations, we find something interesting that challenges many modern assumptions about what constitutes healthy or natural sleep. Studies of the Hadza in Tanzania, the San of southern Africa, and several other traditional populations have found that these groups do not necessarily sleep in a single, long block from dusk to dawn, as modern sleep hygiene guidelines typically recommend. Instead, they tend to sleep in patterns that include periods of wakefulness during the night, during which individuals interact socially, move around, or simply rest without sleeping before returning to sleep. Their sleep onset tends to be several hours after darkness, and their sleep offset tends to be shortly before or after sunrise. These patterns are governed by natural temperature cycles as much as by light, with the lowest nightly temperatures often corresponding to the deepest periods of sleep. The point is not that modern humans should necessarily adopt these exact patterns, which were shaped by environments very different from contemporary ones. The point is that what we call natural sleep, the biological default of the human sleep system when it is operating in the conditions that shaped it, is more variable, more socially embedded, and more temperature responsive than modern discussions of sleep hygiene typically suggest. And understanding this variability is important because it helps us distinguish between the aspects of modern sleep disturbance that represent genuine departures from biological norms, with real consequences for aging, and the aspects that represent simply different expressions of a flexible biological system. The flexibility of the human biological system is, itself, one of the most important features of the human capacity for extended life. The human genome, shaped by millions of years of life in variable and unpredictable environments, did not encode a single, rigid program for development, maintenance, and aging. It encoded a set of conditional programs that respond to environmental signals by adjusting their output in ways that are generally adaptive for the conditions being experienced. When food is scarce, for example, the body shifts resources away from certain forms of cellular maintenance and toward the immediate imperatives of survival at a long-term cost to the pace of aging. When food is consistently abundant, the body has more resources available for maintenance and repair, with generally positive effects on longevity, though this relationship is complicated by the fact that chronic excess of certain nutrients has its own negative effects on cellular health. This conditional character of the human aging program means that the lifespan of any individual human is not simply a function of their genetic endowment. It is a function of the interaction between their genetic endowment and the environmental conditions they experience across their entire life. The same genome, expressed in different environmental conditions, can produce dramatically different patterns of aging. This has been demonstrated repeatedly in animal models, where manipulation of diet, activity levels, temperature, social conditions, and other environmental variables can change the lifespan of genetically identical animals by factors of 2 or more. And while the human case is more complex, involving genetics, culture, and individual behavior in ways that are not fully disentangled, the basic principle holds. Environment matters enormously for how the human body ages, and it matters in ways that are deeply rooted in the evolutionary history we have been exploring. One of the most striking demonstrations of the role of social environment in human aging comes from studies of social isolation and its effects on biological markers of aging. Researchers have found that chronically socially isolated individuals show higher levels of systemic inflammation, shorter telomeres, reduced immune function, and other biological signatures of accelerated aging compared with socially well-connected individuals of similar age and health status. These differences are not trivial. They're large enough to translate in epidemiological studies into measurably higher rates of chronic disease and a shorter lifespan. Social isolation, by the best available estimates, poses a mortality risk comparable in magnitude to smoking roughly 15 cigarettes per day. This finding, which has been replicated in multiple studies across different populations and using different measures of both social isolation and biological aging, makes a great deal of sense in light of the evolutionary story we've been exploring. The human body was designed across millions of years of selection in small, intensely social groups to expect the presence of others. Not just any others, but specific, familiar others. People whose voices and smells and behavioral patterns were as well known to the body's threat assessment systems as the body's own heartbeat. The absence of this social fabric is not for the human body simply a psychological deficit. It is a signal of danger, of exposure, of vulnerability, and the body responds to it the way it responds to any signal of threat, with the sustained activation of stress hormones and inflammatory responses that were adaptive in the short term, when they motivated the individual to seek social reconnection, but that are deeply damaging when they become chronic, as they do when social isolation is a persistent feature of an individual's life rather than a temporary state. This is why the dismantling of traditional community structures in industrialized societies, the atomization of social life that has accompanied urbanization and economic modernization, represents not only a cultural loss but a biological one. The conditions that shaped the human capacity for slow aging were not just physical, they're just social. And the social conditions that supported human longevity in the ancestral environment, the dense networks of mutual obligation, shared history, intergenerational knowledge transfer, and daily face-to-face interaction, are conditions that many people in modern societies experience only partially, if at all. But this observation should not lead us to a kind of nostalgia that is as historically uninformed as it is practically useless. Traditional societies were not uniformly characterized by warm, supportive community life. They had their conflicts, their exclusions, their hierarchies and cruelties. The social bonds that supported longevity in ancestral environments were not always freely available to all members of the group equally. Women, in many traditional societies, operated within social structures that limited their autonomy in significant ways, even as those structures also provided forms of support and connection that modern societies often fail to replicate. The elderly could be, in desperate times, abandoned or neglected when their care became more than the group could sustain. The picture of ancestral social life that emerges from anthropological research is not one of idyllic communal harmony, but one of complex, tension-filled, often difficult social relationships that were nevertheless characterized by a degree of sustained mutual investment and social embeddedness that differs markedly from the conditions of modern individualism. What the research on social aging tells us is not that we need to recreate the specific social structures of the ancestral past, but that the human body has a genuine biological need for certain kinds of social experience, needs that were reliably met in the ancestral environment, and they are often inadequately met at the modern one, and thus meeting those needs as fully as possible in whatever forms are available and meaningful in contemporary life is one of the most important things an individual can do for the pace of their own biological aging. The same logic applies to the relationship between physical activity and aging. The human body does not simply tolerate moderate physical activity, it requires it for optimal functioning. The biological systems that maintain cardiovascular health, muscle mass, bone density, metabolic efficiency, immune function, and cognitive performance are all activated and calibrated by regular physical exertion. In the ancestral environment, this activation was automatic, built into the daily requirements of survival. Food had to be found by walking and sometimes running across large distances. Water had to be carried from wherever it could be found. Children had to be carried when they were small and chased when they were larger. Shelters had to be built and maintained. Fire had to be gathered and tended. The daily energy expenditure of a hunter-gatherer was substantially higher than that of a typical modern desk worker, even accounting for differences in body size. Research on physically active populations and on the effects of structured exercise programs in previously sedentary individuals has found that regular moderate physical activity activates a broad suite of biological maintenance programs. It reduces systemic inflammation. It promotes the growth of new blood vessels in the heart and brain. improving the delivery of oxygen and nutrients to tissues that are particularly vulnerable to the decline of these supplies with age. It stimulates the production of a protein called brain-derived neurotrophic factor, which supports the health, growth, and connectivity of neurons, and appears to be one of the primary mechanisms by which exercise protects against cognitive decline. It activates cellular cleanup programs called autophagy, which remove damaged proteins and organelles from cells and are increasingly recognized as central to the maintenance of cellular health across a long lifespan. And it promotes a pattern of gene expression in muscle cells that supports the maintenance of muscle mass, strength, and metabolic efficiency in ways that slow the decline of physical function that characterizes biological aging. None of these effects requires extreme athletic performance. The research consistently finds that the relationship between physical activity and the pace of biological aging follows a curve in which the largest benefits accrue in the transition from sedentary to moderately active, with diminishing additional returns at higher levels of exertion. Walking, one of the most fundamental human behaviors, turns out to be one of the most powerful interventions for slowing biological aging, not because of any single mechanism, but because of the breadth of biological systems that moderate walking activates. And this makes sense, given that walking was the primary mode of movement for most of human evolutionary history, and that the biological systems associated with aging were calibrated to expect this particular kind of mechanical stimulation. The diversity of diet, as we have noted earlier, plays a similarly foundational role in human aging, and exploring this connection in greater depth reveals some of the most fascinating dimensions of the relationship between our evolutionary past and our biological present. The human digestive system, and particularly the ecosystem of microorganisms that inhabits it, evolved in an environment of extraordinary dietary variety. Recent archaeological and anthropological studies have found that hunter-gatherer diets in different environments included anywhere from dozens to several hundred species of plants over the course of a year, along with a variety of animal foods, fungi, and insects. This dietary diversity is not simply a cultural artifact or a matter of preference. It reflects a genuine biological need, because the gut microbiome requires the exposure to a wide variety of complex fibers and other plant compounds in order to maintain the diversity of microbial species that is associated with healthy gut function. When dietary diversity decreases, as it has dramatically in the context of modern agricultural diets, and more dramatically still in the context of ultra-processed food consumption, the diversity of the gut microbiome decreases as well. And a less diverse gut microbiome is a less functional one, less able to modulate the immune system appropriately, less able to produce the short-chain fatty acids that nourish the cells of the intestinal lining, less able to synthesize certain vitamins and neurotransmitter precursors, and more prone to the low-grade inflammatory state that is associated with accelerated biological aging. This connection between dietary diversity, microbiome diversity, and the pace of aging is one of the most active areas of current research in the biology of longevity, and the findings are consistently pointing in the same direction. Populations that consume more diverse diets, particularly diets rich in a variety of plant foods, show healthier gut microbiomes, lower levels of systemic inflammation, and in many cases slower biological aging, as measured by genomic markers. This is not a simple relationship, and is complicated by many other factors including genetics, lifestyle, and environmental exposures. But the basic pattern is robust. The human body ages more slowly when it receives the diverse nutritional inputs that the gut microbiome and the body's maintenance systems were shaped for millions of years to expect. The relationship between sunlight and aging is another dimension of this story that deserves careful attention. For nearly all of human evolutionary history, humans spent their waking hours outdoors, exposed to the full spectrum of solar radiation that reaches the Earth's surface. This exposure served multiple biological functions, the most obvious of which is the synthesis of vitamin D in the skin, but which also includes the entrainment of circadian rhythms through the action of morning light on retinal photoreceptors, the synthesis of nitric oxide in the skin through ultraviolet exposure, with effects on cardiovascular health and blood pressure, and the modulation of mood and alertness through the effect of light on serotonin and other neurotransmitter systems. The modern pattern of spending most waking hours indoors under artificial light that differs from natural sunlight in both its spectral composition and its intensity represents another dimension of the mismatch between the conditions that shaped human biology and the conditions in which many modern humans actually live. Artificial indoor lighting is typically far dimmer than daylight during the hours when the body expects to receive bright light signals that entrain its circadian rhythms, and far brighter than what the body expects in the evening hours when the biological preparation for sleep should be underway. This disruption of the light environment contributes to circadian rhythm disruption, which has downstream effects on sleep quality, immune function, metabolic health, and ultimately on the pace of biological aging. Research on populations that live with greater exposure to natural light, whether through outdoor work, outdoor recreation, or geographic circumstance, consistently finds healthier circadian rhythms, better sleep quality, lower rates of certain mood disorders, and in some cases markers of slower biological aging compared with populations that live primarily under artificial illumination. Again, the body is responding to the presence or absence of the environmental signals it was shaped to expect, and the response has real consequences for how it ages. All of these threads, the social, the physical, the dietary, the photonic, the microbial, come together in a picture of human aging that is more complex, more contextual, and more profoundly shaped by evolutionary history than any simple biological account would suggest. The human lifespan is not simply a property of the human genome, it is a property of the interaction between the human genome and a particular set of environmental conditions that were specified over millions of years of evolutionary history and that continue to shape the body's aging trajectory in real and measurable ways in every individual human life. Understanding this is important not only for practical reasons, not only because it suggests ways of living that might support healthier aging, but for deeper philosophical reasons as well. It reminds us that we are not isolated biological entities, that we are embedded in webs of relationship, physical and social and ecological, that shape us from the inside out. The pace at which we age is partly a function of the company we keep, the food we eat, the light we see, the distances we walk, the soil we touch, the air we breathe, the stories we tell, and the meaning we find in our days. These are not peripheral factors, they are, in the deepest evolutionary sense, central ones. And perhaps most importantly, this understanding invites a certain humility about the modern project of extending human life through technological intervention. We already possess in our own biology the most sophisticated longevity technology that has ever been evolved. The human genome contains the instructions for building a body that can maintain itself in excellent functional condition for 7, 8, 9 decades, given the right inputs. The challenge is not primarily to create new longevity technology through medicine or biotechnology, though such efforts have their place. The challenge is to provide the ancient longevity technology that we already carry within us, with the conditions it was designed to work in, and meeting that challenge is less a matter of scientific innovation than of cultural wisdom, of finding ways to live in our modern world that are more congruent with the deep biological needs that evolution has written into our bodies over millions of years of patient, incremental, improbable design. This is not a counsel of perfectionism. No human life, ancient or modern, perfectly replicates the conditions under which the human aging program evolved. Hunter-gatherers got sick. They were hungry. They were cold. They were afraid. They lost people they loved and experienced grief that damaged them in ways we are only beginning to understand. They were not perfectly healthy, perfectly active, perfectly connected, perfectly nourished beings. They were ordinary human beings doing their best in difficult circumstances, just as we are. The difference is not that their circumstances were perfect, but that in certain fundamental respects, the demands of their daily lives provided their bodies with inputs that the biological machinery of slow aging needed and expected. What we can take from this evolutionary story is not a prescription, but a direction, a sense of what the body is looking for, of what signals it interprets as invitations to maintain and repair and sustain itself, and of what signals it interprets as reasons to divert those maintenance resources elsewhere. And armed with that sense, we can make choices, imperfect choices, compromised choices, choices constrained by the realities of modern life, that nevertheless move us incrementally toward the conditions under which our extraordinary biological inheritance is most fully expressed. The body wants to move, not to perform, not to compete, not to achieve some arbitrary metric of athletic excellence. It wants to move in the sustained, varied, moderate ways that kept our ancestors alive and fed and capable across long lives in difficult terrain. It wants to walk across varied landscapes and carry things and climb and squat and lift and rest and walk again. The body wants to eat variety, not any particular cuisine, not any particular dietary philosophy, but a genuine diversity of plant foods in quantities and combinations that maintain extraordinary community of microorganisms that lives within it, and that is, in a very real sense, part of it. The microbiome is not a passenger in the human body. It is a partner, and treating it as such, feeding it the diversity it needs to remain diverse and functional, is one of the most powerful things a person can do for the pace of their own aging. The body wants to sleep in synchrony with the natural world, not perfectly, not rigidly, but in rough alignment with the cycles of light and dark that have governed the biological clocks of all life on Earth for billions of years. It wants to see morning light and to experience the gradual dimming of the evening as a signal that the time for rest is approaching, rather than as an arbitrary decision to be made in the presence of screens and artificial illumination. The body wants company, not the company of performance or obligation, not the shallow sociality of digital platforms, but the company of people who are genuinely known and genuinely nowhere, who share history and vulnerability and care. The body wants to be touched, and to touch, to hear familiar voices, to participate in the small daily rituals of mutual acknowledgement and support that bind human groups together and that were, for most of human history, as automatic as breathing. And the body wants meaning. This is perhaps the hardest to quantify. the hardest to study, the hardest to provision in a modern world that has, in many ways, become expert at providing stimulation while starving meaning. But the evidence that a sense of purpose and meaning matters for biological aging is consistent and compelling. Populations with high levels of what researchers call ikigai, a Japanese concept roughly translatable as reason for being, show lower rates of chronic disease and longer lifespans than populations without this quality. Individuals who report a strong sense of purpose in their lives show slower biological aging as measured by telomere length and inflammatory markers than those who report low purpose, even after controlling for other relevant factors. The body, it seems, is listening to the story the mind is telling itself about why it is here and what it is for, and it is aging faster or slower depending on the quality of that story. This is a striking finding, and it is one that connects the evolutionary story of human aging to the deepest questions of human existence. The human capacity for slow aging evolved in a context in which every individual's contribution to the group was visible and necessary. In that context, purpose was not a luxury or an aspiration. It was a daily reality, woven into the fabric of every interaction and every activity. Finding food had purpose, caring for children had purpose. Teaching a young person how to track an animal or identify a medicinal plant had purpose. Maintaining the social bonds of the group had purpose. Telling the stories that preserved the group's history and identity had purpose. And the body, shaped by millions of years of living in this purposeful context, appears to have evolved to expect purpose as a basic input, in the same way that it expects physical activity and dietary diversity and social connection and adequate sleep. When we live without purpose, or with a purpose so small and so disconnected from the lives of others that it provides no real sense of meaning, the body registers this absence in ways that have measurable biological consequences. The same stress hormones that are activated by social isolation, by chronic sleep deprivation, by physical inactivity, are activated by purposelessness. The same inflammatory pathways that accelerate aging in the presence of other chronic stressors are activated by the chronic low-grade psychological stress of a life without apparent meaning. The body is not indifferent to the story the mind is living. It is profoundly responsive to it. This is perhaps the deepest and most surprising implication of the evolutionary story of human aging, that the biology of longevity and the philosophy of a good life are not separate concerns to be addressed by separate disciplines in separate domains. They are the same concern approached from different angles. The conditions that support slow biological aging are, in very large measure, the conditions that support a rich and meaningful human life. Genuine connection with others, meaningful contribution to something beyond the self, regular physical engagement with the world, nourishment from real food in real variety, rest aligned with the natural rhythms of light and dark, and the experience of being known and valued in a community of mutual obligation and care. This convergence is not coincidental. It is the product of millions of years of evolution that shaped the human body for a particular kind of life, a life that was, for most of human history, the only kind of life available. And the fact that this kind of life also happens to produce slower biological aging is not separate from the fact that it was also in many ways a deeply human and deeply satisfying kind of life. The evolutionary forces that extended the human lifespan were not rewarding discomfort or sacrifice for its own sake. They were rewarding the things that made human social life work, the behaviors and commitments and relationships that held groups together and allowed knowledge to accumulate and be transmitted, and made it possible for the extraordinary social experiment of human civilization to unfold. We are the inheritors of that experiment. We carry within us the biological traces of every ancestor who lived long enough to matter to the people around them, who was valued enough to be fed and protected and taught, whose knowledge was rare enough to be worth preserving in a living body, rather than being lost to the indifferent fact of an early death. Every decade of lifespan that distinguishes us from our chimpanzee cousins is a decade that was, in some ancestral environment, earned by someone who had something to offer, something to teach, something to give. And the body that allows us to live those decades, to remain capable and engaged and contributing well past the ages at which our evolutionary relatives are already in decline, is the body that millions of years of selection built for that purpose. To live fully within that body, to give it the conditions it was designed to work in, to honour the extraordinary biological inheritance that every cell of it represents, is not a project of self-optimization or longevity hacking. It is something simpler and older and more fundamentally human than that. It is the project of living the way that living beings with our particular biology were designed to live, in connection and in motion and in meaning and in care, for as long as this brief and remarkable possession of a human body allows. The night deepens. Somewhere in the darkness outside, the world continues its ancient processes, its cycles of growth, decay, its slow geological shifts, its patient biological experiments. The body you carry, older by far than its individual years, wiser than any mind can fully comprehend, begins its nightly work of repair and consolidation and preparation for another day. Let it do this work. Let it have the rest it was designed for. Let the old rhythms hold. Sleep now with the knowledge that the machinery of your aging is one of the most remarkable things in the history of life, and that it is even now, even in this imperfect world, doing its very best to give you more time. There is a concept in anthropology, sometimes called the ecology of knowledge, and it refers to the way that, in small-scale human societies, knowledge is not simply stored in individual minds, but is distributed across the social network, embedded in relationships, practices, rituals, and landscape, maintained through constant use and repetition in the context of daily life. No single individual in a hunter-gatherer band knows everything that the group collectively knows. The elder woman knows the medicinal plants and the seasonal patterns of roots and fruits. The experienced hunter knows the behavioral rhythms of the game animals and the geography of the terrain across many days' travel in every direction. The skilled craftsperson knows the particular properties of local stone and fibre and wood, which materials are suitable for which purposes, how to read the quality of a piece of flint from the sound it makes when struck, how to select a length of wood for a bow that will be strong without being brittle. The midwife knows the signs that distinguish a normal from a dangerous labour. The storyteller knows the narratives that encode the group's history and values, and that provide the framework within which individual experiences can be interpreted and understood. This distributed knowledge is the real resource that makes human social life work, and it is a resource that depreciates catastrophically with the death of any of its holders. This is why the loss of an experienced elder in a small hunter-gatherer band was not simply a social tragedy, but an informational one, potentially fatal to the group in ways that the loss of a younger, physically capable but less experienced individual was not. And this is why selection for longevity in our species was not a simple or straightforward process, but one deeply entangled with the evolution of social complexity itself. As hominins evolved toward greater cognitive complexity, toward more sophisticated language, more elaborate tool use, more complex social structures, and more extensive home ranges, the amount of accumulated knowledge that experienced individuals carried grew correspondingly. And as that accumulated knowledge grew in its depth and breadth and practical importance to group survival, so too did the selective value of the individuals who carried it. This is a positive feedback loop of remarkable power. Cognitive complexity selects for greater longevity, because complex knowledge takes longer to accumulate and is more valuable once accumulated. And greater longevity selects for greater cognitive complexity, because longer-lived individuals can accumulate more complex knowledge and can sustain the extended social learning networks through which complex knowledge is transmitted across generations. This feedback loop is, in all likelihood, one of the primary engines that drove the simultaneous expansion of the human brain and extension of the human lifespan over the past 2 million years. The two processes are not independent phenomena that happened to occur in the same lineage. They are deeply interconnected aspects of a single evolutionary process, the bootstrapping of human social complexity into ever higher levels of organization and capability, driven by the extraordinary selective value that complex knowledge creates in a species that has bet its evolutionary future on intelligence and social cooperation rather than on physical prowess or simple behavioral specialization. One of the most powerful pieces of evidence for this interpretation comes from studies of brain volume and lifespan across the primate order. When researchers plot the maximum lifespans of different primate species against their relative brain volumes, correcting for body size, they find a strong positive correlation. Primates with relatively larger brains tend to live longer than primates with relatively smaller ones. This relationship holds across dozens of primate species and is robust to multiple analytical approaches. And it makes biological sense, given the argument we've been developing. Larger brains are, in evolutionary terms, expensive investments that take a long time to pay off, and the payoff is maximized when the individual who possesses that large brain is able to apply it across a long life of accumulated experience rather than died before the investment has had time to generate its full return. The human brain, the largest relative to body size of any primate, and correspondingly the most expensive to build and maintain, represents the extreme end of this relationship. And the human lifespan, the longest of any primate, represents the corresponding extreme of the selection pressure for maximizing the return on that extraordinary cognitive investment. Together, they constitute what is arguably the most dramatic example of coevolution between cognitive and somatic traits in the history of the primate order. A partnership between a brain that can learn almost without limit and a body designed to give that brain the time it needs to put its learning to use. From an evolutionary perspective, the timing of this coevolution is deeply significant. The most dramatic increases in human brain volume relative to body size occurred in the genus Homo over the past 2 million years, approximately coincident with the development of stone tool technology, with the first evidence of controlled fire use, and with the emergence of the social and cooperative behaviours that characterise anatomically modern humans and their close ancestors. This was not a period of gradual, smooth change. It was, by the standards of evolutionary biology, a rapid and dramatic transformation, driven by the extraordinary power of the feedback loops we have been describing. And across this period, one of the things that was changing was the pace of aging. The earliest members of the genus Homo, roughly 2 million years ago, show skeletal evidence of aging patterns that are intermediate between those of earlier Australopithecines and those of modern humans. Their teeth wore down faster. Their joints showed signs of degenerative changes at younger biological ages. Their bones show patterns of mineral density loss that suggest a somewhat faster pace of somatic decline than is typical in modern human populations. As the lineage evolved toward anatomically modern humans, over the course of roughly 2 million years, the pace of this somatic decline appears to have slowed, reflected in skeletal evidence from older individuals who show signs of having survived injuries and illnesses that would likely have been fatal in earlier, less robust bodies, and in the presence in archaeological sites of very old individuals whose survival required the sustained support and care of the group. The archaeological record of human aging is itself a remarkable document, though one that must be read carefully, because the evidence is fragmentary and the inferences it supports are necessarily qualified. Paleoanthropologists can estimate the age at death of skeletal individuals through a variety of methods, including the examination of dental wear, the degree of fusion of bone growth plates, the density and structure of bone tissue, and in some cases the pattern of degenerative changes in joints. These methods are imprecise, particularly for old individuals, and they provide estimates rather than exact ages. But across the entire Paleolithic record, from the earliest members of our genus to anatomically modern humans of the late Pleistocene, there is a clear trend toward longer survival and toward more frequent evidence of individuals who reached what we might call old age, individuals who are not only surviving to older ages but doing so within social groups that appear to have been providing them with care and support. The evidence for care of the elderly in the Paleolithic is among the most moving and philosophically significant findings in all of paleoanthropology. At multiple sites, researchers have found skeletal individuals who survived for years with disabilities or injuries that would have made independent survival impossible. A Neanderthal individual from Shanidar Cave in Prague, known as Shanidar I, and dating to roughly 50,000 years ago, shows evidence of multiple severe injuries, including a withered arm, damage to one eye that likely caused blindness, and injuries to the lower limbs that would have significantly impaired mobility. This individual survived to an advanced age by Neanderthal standards, in conditions that would have required sustained support from the group. Similar evidence of care for injured or disabled individuals has been found in multiple other Paleolithic sites, suggesting that the practice of caring for those who cannot fully care for themselves is ancient, predating modern humans, and deeply embedded in the social fabric of the human lineage. This evidence of ancient care is not simply a heartwarming story about the kindness of our ancestors. It is a crucial piece of the evolutionary argument for the origins of human longevity, because the existence of care for disabled and elderly individuals implies a social recognition of their value, a judgment, made individually and collectively by the members of the group, that the continued presence of the disabled or aging individual was worth the cost of their support, and in an environment of limited resources, where every calorie was earned through labour and every individual was expected to contribute to the group's survival, that judgment was not made lightly. It was made because the value of experienced individuals, of people who had accumulated knowledge and social relationships and practical skills over long lives, was real and recognized, and significant enough to justify the investment of care. The willingness to invest in the survival of older and less physically capable individuals is, in this light, not a departure from the logic of natural selection, but an expression of it. In a species where accumulated knowledge is the primary survival advantage, and where that knowledge exists only in the minds of the individuals who have taken the time to accumulate it, caring for knowledgeable elders is one of the most evolutionarily rational things a group can do. And the fact that this care also reflects genuine emotional attachment, genuine love and loyalty and obligation, does not make it less rational. It makes the evolutionary logic more powerful, because it means that the care was maintained not only when it was immediately and obviously beneficial, but across the full range of circumstances that characterize the variable and unpredictable life of a hunter-gatherer band. The emotional dimension of human longevity is one that is rarely discussed in scientific accounts, but it may be one of the most important. The particular pattern of emotional attachment that characterizes human social life, including deep and sustained bonds between parents and children, between siblings, between life partners, between close friends, and between grandparents and grandchildren, creates the motivational infrastructure that sustains the long-term social investments that human longevity depends upon. It is not simply that older humans are kept alive because the group recognizes their cognitive value. It is that they are loved in ways that make their loss genuinely painful and their continued presence genuinely desired, and that this love motivates the kind of sustained care and support that allows older individuals to survive challenges that would otherwise be fatal. This emotional architecture of human social life did not arise independently of the cognitive and demographic changes that produced the human lifespan. It co-evolved with them in a mutually reinforcing process in which the deep emotional bonds of human family and friendship created the social conditions that made longevity valuable, and the value of longevity created selection pressure for the deepening and extending of those emotional bonds. The result is the extraordinary richness of human emotional life, the capacity for love and grief and loyalty and obligation that extends across time and across the generations in ways that are, among primates, uniquely human. And perhaps this is ultimately the most profound thing that the evolutionary story of human aging has to tell us, that the extraordinary biological machinery of slow human aging did not evolve in isolation from the rest of human nature. It evolved together with language, with culture, with the capacity for deep emotional attachment, with the love of stories and the making of meaning, with the extraordinary human ability to care for others across time. These things are not accidental accompaniments to the human capacity for long life. They are its evolutionary partners, the social and emotional infrastructure that made long life worth selecting for, and that continues to be, in the deepest biological sense, what long life is for. We age slowly as humans because we were worth keeping, because the things we carry in our minds and our hands and our hearts across long decades of living are rare and valuable and necessary to the people who love us and depend on us. Because the species that became us discovered, across millions of years of painful and glorious experiment, that the oldest among them were not burdens to be discarded, but wells of experience to be drawn from, teachers to be learned from, protectors to be sheltered under, storytellers whose voices held the group together in the dark. We aged slowly because we were designed for each other, and that is a thing worth knowing in the quiet hours of the night, as the body does its ancient work and the mind grows still, and the long story of what we are continues its patient unfolding in the dark. Let the silence hold you now. Let the slow work of living continue. You are older than you know, and younger than you feel, and exactly as remarkable as the 2 million years of selection that shaped this moment of your rest. Sleep well. There is one more thread in this story that deserves careful attention before we let it rest, and it concerns the question of what exactly happens to the aging body during sleep, and why sleep has been, for most of human evolutionary history, the most important daily act of biological maintenance that the human organism performs. This question is not peripheral to the story of human aging. It is central to it, because the extraordinary biological maintenance that keeps the human body functioning across 8 or 9 decades of life happens largely during the hours that we typically regard as lost time, the hours of unconsciousness and dream and slow restoration that constitute sleep. When the body sleeps, the brain does not simply rest. It begins a series of processes that are, if anything, more energetically demanding than many waking activities. The neurons that were active during the day, forming and strengthening connections in response to experience and learning, are now undergoing a process of selective pruning and consolidation, retaining the connections that are most important and allowing others to weaken. This process, which appears to require sleep specifically and cannot be adequately replaced by quiet waking rest, is one of the primary mechanisms by which learning becomes permanent memory, by which the experiences of the day are integrated into the existing architecture of knowledge and understanding. But beyond memory consolidation, sleep activates the glymphatic system in ways that are only beginning to be appreciated in their full significance. The glymphatic system, which functions poorly or not at all during wakefulness, pumps cerebrospinal fluid through channels in the brain with remarkable efficiency during deep sleep, clearing away the metabolic byproducts of daytime neural activity. These byproducts include proteins that, if allowed to accumulate over years and decades, form the characteristic deposits associated with neurodegenerative diseases. The brain's nightly cleaning process is, in a very real sense, one of the most important anti-aging behaviors available to the human body, and it occurs only during sleep. Beyond the brain, the sleeping body is conducting repairs and maintenance throughout every organ and tissue. Growth hormone, which despite its name is important throughout life for the maintenance and repair of muscle, bone and other tissues, is secreted primarily during the early stages of deep sleep. The immune system during sleep shifts from its defensive surveillance mode into a more active mode of cellular repair and the consolidation of immune memory, the process by which the immune system retains information about past pathogens so that it can respond more rapidly and effectively to future encounters. Inflammatory processes that were activated during the day in response to the normal cellular stresses of waking life are modulated during sleep in ways that prevent them from becoming chronic and damaging. And the hormonal rhythms that govern metabolism, appetite, and energy storage are reset during sleep in ways that influence how the body manages energy for the entire following day. All of this biological activity during sleep depends on the adequate duration and appropriate architecture of sleep, meaning the proper sequence and proportion of different sleep stages, from the light stages through which we enter sleep to the deep, slow-wave stages during which the most intensive physical repair occurs, to the rapid eye movement stages during which memory consolidation and emotional processing are most active. And the architecture of sleep is itself regulated by the circadian rhythm, the approximately 24-hour biological clock that governs almost every physiological process in the body, and that is primarily set by exposure to light at appropriate times of day and night. In the ancestral environment, this circadian regulation of sleep architecture was largely automatic. The gradual brightening of dawn naturally stimulated the release of cortisol, the hormone that promotes alertness and prepares the body for the activities of the day. The gradual darkening of evening naturally stimulated the release of melatonin, the hormone that promotes drowsiness and initiates the preparation of the brain and body for sleep. The temperature drop of the night, which in the environments where the human species evolved was often considerable, further promoted a sleep onset and the maintenance of the deeper sleep stages, and the absence of artificial stimulation during the hours of darkness meant that once sleep began, it was generally sustained through the biological rhythms of the night, without significant interruption. The modern disruption of these conditions is one of the most consequential and least widely appreciated aspects of the mismatch between the ancestral environment and the contemporary one. Electric lighting, which allows and indeed encourages wakefulness at hours when the circadian system is preparing the body for sleep, has compressed the sleep period of many modern populations below the levels that the biology of sleep maintenance and repair requires. The proliferation of screens emitting short wavelength blue light, which is particularly effective at suppressing melatonin secretion, has further disrupted the hormonal preparation for sleep, and the increasing prevalence of sleep disorders from sleep apnea to chronic insomnia suggests that the conditions under which the human sleep system evolved are not reliably replicated by the conditions under which many modern humans attempt to sleep. The consequences for aging are significant. Research on sleep deprivation, both acute and chronic, consistently finds that it accelerates many of the biological processes associated with aging. Telomere shortening is faster in individuals who chronically sleep fewer than 7 hours per night. Inflammatory markers are elevated after even a few nights of inadequate sleep. The clearance of amyloid proteins from the brain during lymphatic activity is reduced. Immune function is impaired. Metabolic regulation is disrupted in ways that promote insulin resistance and the accumulation of visceral fat. And cognitive function, including the domains of memory, attention, and executive control that are among the first to show signs of decline in normal aging, is impaired by sleep deprivation in ways that in the short term are reversible, but that in the long term, if chronic, may contribute to lasting neurological changes. This is not an argument for anxiety about sleep, which will be counterproductive in the extreme. It is an argument for understanding sleep as what it actually is, not lost time, not a concession to biological weakness, but one of the most fundamentally important things a human body does, one of the primary ways in which the extraordinary biological machinery of slow human aging does the work it was designed to do. Honouring sleep, protecting it, creating conditions that allow it to unfold in accordance with the biological rhythms that evolution shaped, is one of the most direct and effective ways of supporting the biological inheritance that makes the human lifespan possible. And now, having followed the threads of this story as far as they can take us tonight, having traced the arc from the African savanna 2 million years ago to the molecular mechanisms of cellular repair, from the evolutionary logic of grandmother's generosity to the biology of the lymphatic system, from the social fabric of hunter-gatherer life to the neurochemistry of circadian rhythms, we arrive at the simplest and most important place of all, the end of a day, the beginning of a night, and the body's quiet invitation to do what it was designed to do in these hours, The story of human aging is, in the deepest sense, a love story. Not a romantic love story, or not primarily, though the bonds of romantic love are certainly part of it. It is the love story of a species that discovered, across millions of years of living and dying and learning, that the most valuable thing any of its members could do was to stay alive long enough to matter deeply to the people around them, long enough to teach what they had learned and share what they had gathered and hold what they knew and love what they loved and be for as long as possible themselves. You are the current iteration of that story. The body you will carry into sleep tonight is the product of every ancestor who lived long enough to love someone, who was loved enough to be cared for, who cared enough to stay and teach and give. It is a body that contains in its cells and its systems and its slow biological rhythms the accumulated investment of millions of years of selection for precisely this, for the capacity to be here and to matter and to rest and to wake again. Let it rest now. The story will continue in the morning as it always has, as it was always going to, as it is designed across all those unimaginable years of patient selection to do. Close your eyes. The darkness is old and kind and full of the breathing of all the sleeping generations that brought you here. You are home. Sleep now.